Yeah, so neurotransmitters. So we're going to talk about some specific neurotransmitters, talk a little bit about different effects that a neurotransmitter can have on a target tissue. And yeah, so we'll go from there. So let's start out with just talking about all the different cells that the nervous system can control, okay, or the different kind of tissues that could be a target tissue for a neuron, right? So neurons are going to be connecting to something, releasing something, and controlling that, right? So these are kind of the options here in the letters there. And so just take a second and match up which types of neurons would go to each of these, right? So you're taking those letters and putting them underneath one of those, one or more of those three bullet points there, okay? And then on your clickers, go ahead and send in the one that would be related to interneurons there, okay? Chat with your team about this, send those in. It's like about 13 people didn't get the memo that we still had school this week. I'm not sure how that was confused. Just kidding. How many people have another class that was canceled this week? Yeah, most of you guys, yeah. And therein lies the problem. It's not just the students trying to get out of it. You guys almost have a full team, not quite. This one.
There's one of us missing, all of us are missing.
Oh, wow. It's a true team.
Yeah.
Team effort. Yeah, I love it. I have a full team gone over here. Wow. Team effort for, yeah, we're not showing up. Wow. Yeah, totally. All right, send that in on your clickers there. I'll leave it open for a sec so you can continue to do that. I think about 5 or 6 more of you still need to send that in. Okay, so which letters did you put under autonomic neurons? An axon of an autonomic neuron, which ones could it go to here? Could be A, yeah, could be a gland, for sure. What else? Yeah, could be smooth muscle, for sure. D, could be cardiac muscle, for sure. Yeah, anything else? That's pretty much it, yeah, okay. Somatic motor neuron, which one? B, yes, oops. So just, yeah, just skeletal muscle there, striated muscle. Okay, and then sensory interneuron, what'd you put there? E, yeah, so interneuron just implies that it goes from neuron to neuron, right? Neuron to neuron communication would be interneuron. Okay, good. And that's what most of you put, about 75% of you put that, so good. Okay, questions about that at all? Okay, so these are all the types of cells or tissues that the nervous system interacts with, which means that these are the type of cells that the nervous system controls, okay? And so there's two different types of synapses that can exist between a neuron and one of those cells, okay? So it can be an electrical synapse, okay, which that means there's no space between the two cells, there's no cleft, okay, that's the space. Instead, there's gap junctions, okay? So that means the cells themselves are connected just directly to each other, or it could mean that the neuron is connected directly to the cell. And so that electrical signal just moves directly from one cell to the other, okay? This helps cells to act kind of as a single functional unit, okay? So some examples of this. This is what happens up in the hypothalamus, one of the important glands up in our brain. It's going to be controlling our endocrine system. We'll get into that next semester. This happens between cardiac muscle cells as well. We can send that signal from one cell to the next without any nervous system needed between the cells. We start it with the nervous system, and then some smooth muscle cells act this way too. where we just have a bunch of cells connected to each other, including neurons connected to those smooth muscle cells, which just can activate them, and that signal just spreads throughout. Okay, that's the much less common version. The much more common version is a chemical synapse. Okay, and so this is where we have the axon kind of spreads out at the base, right? There's in that spread out part, that little bulb at the end of the axon, there's a bunch of vesicles, right? Like little sacs full of neurotransmitters. Okay, and then there's a little space between. That's the synaptic cleft. This is where the neurotransmitter diffuses through. Okay, and then on that target tissue, there's a specialized part of the membrane that is usually heavily folded and has receptors for that specific neurotransmitter. Okay, so that's kind of what we talked about with skeletal muscle, right? The neuromuscular junction is an example of that. Okay, this is the most common thing. This is most neuron to neuron interaction is this way, skeletal muscle this way. So this is the primary way that we would see a neuron interacting with another tissue would be with this type of chemical synapse. Okay. Questions on that at all? All right. Okay. So just take a quick look at this here. Just throw it in your clickers just quickly just to make sure we're on the same page. We got those two examples there. Which one would you say is the chemical one? Check with your teammates real quick just to make sure. All right, send something in if you haven't yet. So yes, most of you, at least about 85% of you are saying B is the chemical synapse, and that is true. With A, we can see gap junctions, right? So those cells are connected directly to each other. Ions can pass from one cell to the next. So that's like the electrical signal just passing from one cell to the next, okay? And then in the chemical synapse, we have this space, right? We have the synaptic cleft, and we're going to be dependent on a neurotransmitter being released here, and then it's going to go bind to this receptor and send the signal that way. Okay, so that's the chemical synapse. So we'll spend most of the time today talking about the chemical synapse and not the electrical one. Okay, questions on those at all? Okay. All right, so as a bit of a review here, we pretty much did these steps already when we talked about the neuromuscular junction. Okay, this would be just very similar to that, but looking at more of a neuron to neuron chemical synapse. Okay, this is going to result in a graded potential, like we did, like we saw in the neuromuscular junction as well. Okay, so there's kind of three slides here that walk you through the steps of those things. So go ahead and yeah, just walk through that with the people on your team. Write in what's going on there. And then you can also answer this clicker question on the fourth slide after this. I'll have that open for that. Okay, so go ahead, walk through all of those steps, write in, fill in what's going on, and answer this clicker question. Do that together. We'll come back together here in a couple minutes.
Yeah.
I guess here, sorry, let's do it this way. I guess I forgot I did have a clicker question right there. So let's do that one first. We'll do this one first, which is a number one, okay? And we'll do that other one in a sec. Say it again. This is what we went over when we did the neuromuscular junction. It's kind of very similar steps to that. So it was back a little ways before all that stuff. Before all that too, it was probably like right when we started the nervous system.
Yeah.
Before that too. It's probably the one before that would be my guess. Or the spinal nerves, maybe.
Oh, like from.
Yeah, it was from the previous section. Yeah. It's called neuromuscular junction, I think. Yeah, this one here. This set of notes here. Yeah, there you go.
Oh,
Yeah, it's a ways back.
I was like that.
Yeah, so neuromuscular junction notes were back in the previous exam section. Nothing's gonna uncover, so you might as well write it down.
Okay. Okay, I was just gonna-
These ones don't uncover either, so you might as well write down your answer.
After the NA channels, then potential.
Star circulated potential. All right, send in a number there for this question, and then we'll start walking through it. Make sure to hit the send button with this type of question. So a couple of you still need to send it in. Okay, let's walk through it together here. I'll leave it open. I think there's at least one more person that needs to hit send still. Yeah, so step one, what did you guys write there? Action potential. Yeah, action potential comes down the neuron, right? So we see the action potential coming down. That's step one. The action potential then passes over these voltage gated calcium channels, right? So step two is calcium rushes into the cell, into the neuron, right? Because of that action potential, it changes the shape of those calcium channels, opens them up, right? Calcium rushes into the cell. So we got action potential comes down, that opens up these calcium channels. Calcium comes into the cell. What does calcium do when it gets in there? Huh? Yeah, you're thinking the muscle contraction one? Yeah. What does it cause? Yeah, causes these guys to move to the surface here, and then they dump their neurotransmitter out into the space. Could be acetylcholine. It doesn't have to be though, right? That's just one of the many neurotransmitters that neurons can release. Okay. So we'll just say in general, it causes the release of the neurotransmitter. Okay. So, oops. Action potential arrives. Okay. Calcium rushes in. Okay. Neurotransmitter release. Okay. And then number four, neurotransmitter, we'll say NT binds to receptor, right, on the target tissue. Okay, so neurotransmitter binds to the receptor there. Okay, and if you look up here, this is where we're zoomed in on, right? So this is an axon of one neuron. That's this axon here, right, is interacting with the soma of another neuron. So that's kind of what we're looking at here, right? So this neuron is going to be affecting this neuron, right? And there'll be a couple different options for how it affects it. It could lead it towards an action potential, or it could lead it away from an action potential, right? So it could try to inhibit that neuron, or it could try to activate that neuron, depending on what type of synapse it is, okay? Okay, so questions on these first four steps here? Okay, so then after the neurotransmitter binds, it's going to cause a graded potential. So we get a graded potential. Okay, that's what we're seeing there. Okay, so most of you put 4 for the answer of when the neurotransmitter has its effect on it. That's fine, because on that one it looks like that's when the neurotransmitter binds to it. But 5 would probably be where you can really see it in these images, right? Because you can kind of see this little graded potential thing shown by these arrows. Okay, so we got that. All right, and then the last step is the neurotransmitter is removed in one way or another. Okay, it can be taken back up into the neuron that released it. That's called reuptake. Okay, that's an important term for some of the neurotransmitters that we'll talk about. We'll talk about drugs that affect the reuptake of certain neurotransmitters, right? SSRIs, right? Selective serotonin reuptake inhibitors, right? Those are very common antidepressant medication, right? It stops this activity, right? Stops these little pumps from pumping the serotonin back up into the neurons. So serotonin stays out here longer, right? And when serotonin stays out there longer, it has more effects on the target cells that it's binding to, right? Serotonin's a feel-good neurotransmitter, right? So it can kind of help with depressive symptoms by leaving serotonin out there longer, right? So it's just one example. So that's one example of what can happen to end the signal. Another example would be that there's an enzyme there that degrades it. That's what we saw with acetylcholine, right? That acetylcholine esterase that broke down the acetylcholine. That made it stop the signal. And then the third option will be just that the neurotransmitter diffuses out of that space. We stop releasing it, so there's not as much of it, and then some of it just diffuses out, so we don't have that effect anymore, right? So there's kind of 3 different options for how do we stop the signal from that neurotransmitter. One would be we take it back up into the cell that released it. 2 would be we break it down with an enzyme. 3 would be we just let it diffuse from that space. Okay? All right, questions on that? Okay, so quick check here. Go ahead, chat about that with your team and send that in. What's the role of calcium in the release of a neurotransmitter in a chemical synapse? Check with your teammates here real quick. Send in your answer.
Yeah, because that's the same thing.
All right, so send your answer in. About 10 more of you. All right, I'll leave it open for a sec if you're still sending that in. But yeah, so about 90% of you are saying B is the right answer. Yeah, so to stimulate the release of the neurotransmitter. Perfect. That's what it's doing. Okay. All right. So let's talk about the different types of postsynaptic potentials. Okay. So when a neurotransmitter binds to another neuron, it can either excite that neuron. Okay. And in that case, it would cause depolarization on that neuron. This is called an excitatory synapse, and the electrical change that it causes is termed an EPSP, or excitatory postsynaptic potential. Okay, so it just means it excites the cell that it interacts with, means that it moves it towards an action potential. Okay, so an EPSP. And then the other option is an inhibitory synapse, and in that case, the neurotransmitter causes hyperpolarization instead. Right, so instead of going up in the positive direction towards the threshold for an action potential, it goes the other direction. It goes more negative, so it makes it go further away from that threshold for an action potential, so it's less likely for that cell to get excited and to send its own signal, right? So we're inhibiting that cell in one of these inhibitory synapses. This is called an IPSP. Okay, so again, these are graded potentials, and graded potentials can sum on top of each other, right? So if we get a bunch of IPSPs, it pushes us further and further into hyperpolarization away from that action potential threshold, right? So it makes it harder for other neurons that might be interacting with that same one to excite it. If we might have excitatory ones releasing onto a neuron, and then other inhibitory ones releasing onto that same neuron, and it's kind of this back and forth, right? And so the inhibitory ones are pushing it further away from the threshold. Excitatory ones are trying to push it in the direction of excitation up towards the action potential threshold. Okay, so if a bunch of EPSPs happen together, right, then we're going to reach that action potential threshold and we can get an action potential. All right, a bunch of IPSPs sum together, then we're just moving further into hyperpolarization, right? Further away from that action potential threshold. To put this into perspective, the average motor neuron that comes from the spinal cord interacts with 10,000 other axons before it actually gets to your muscle. So that means you picture that one motor neuron going down to my vastus lateralis, right? There's a bunch of them, but just picture one of them. 10,000 other neurons are trying to mess with it before it actually gets to my muscle, right? So there's a lot of inhibition, there's a lot of excitation, right? With that tetanus infection, the reason that people contract so hard isn't because we're overstimulating it, it's because that bacteria actually gets up into the neuron and blocks all the inhibitory signals. That's actually why it contracts so hard with the tetanus contraction, is because we're blocking all of these IPSPs instead. So that just shows you how important it is to kind of smooth out the signal that gets sent on a neuron, just to do something simple like contract a muscle, right? So lots and lots of kind of smoothing and changing the signal before it actually gets to the muscle, right? And that's what we see with a lot of this. Okay, so this is kind of the big picture here, right? We're sending this signal down, right, to another neuron here, and then we can either get an EPSP, right, pushing us further to the threshold, closer to the threshold, or an IPSP here pushing us away from that. Okay, and the things that can cause that are shown here. There's kind of two different types of ion flows that lead to this. Okay, and so we can see this here on the depolarizing side. That's this one here. So this EPSP, pretty much our two options are we can have a sodium channel or a calcium channel. All right, so this could be due to sodium influx or calcium influx. Okay, and so these are different types of channels. They're both ligand-gated ion channels, right? The ligand is the neurotransmitter, right? And that's the key to open or close this door of the ion channel, right? And so in some excitatory synapses, there's sodium channels. In other excitatory synapses, there's calcium channels. They both have a similar effect though, right? When we send a positively charged ion into the cell, then it's going to depolarize it. It's going to move us in this direction, right? The options on the inhibitory side include a ligand-gated potassium channel. Okay, so this would be potassium efflux or chloride influx. All right, so we can see the chloride one here. So there's a chloride, ligand-gated chloride channel. Chloride channel opens up, and the negatively charged ion goes in. Okay, that would push us further in the negative direction, away from the action potential threshold. Same thing with potassium going out, right? Potassium going out pushes us in the negative direction as well. Both of those things would be inhibitory, right? They would push us further away from that action potential threshold. Okay. Questions on that? Yeah, Maddie, go ahead.
Sodium channel. For based on the charges that we look at, or
Do you mean what you're looking for, or like how the protein's structured to allow the different ion through? Yeah, it has to do with the size of the opening and the charges of the amino acids in the opening that kind of allow it through. So yeah, super specific, a lot of them that only allow a single type of ion through, but it's all about the size and the charges of the amino acids on the interior of this thing that would allow it or not allow something to pass through. So, yeah. Good question. Other questions? Okay. And then all of these things are moving with their concentration gradient, right? There's no active transport here. This is all just opening up. So then they can go from high concentration to low, right? With all of these examples. Okay, so so let's talk a little bit about the different types of receptors and channels. And first of all, we'll talk about what types of ligands we have. And the ligands, again, a ligand is a chemical that binds to a receptor. Okay, that's our definition of a ligand. Okay, so the function of it is it allows for one cell to communicate with another when one cell releases one of those ligands. Okay, and there's really three types of ligands in the body. The first one is neurotransmitters, right? And the way these are defined is by three things, well, really by two things. Is what releases it and what it releases it into. Okay, so if a chemical is released from a neuron and it goes into a synaptic cleft, then it's a neurotransmitter. That's it. That's what makes it a neurotransmitter, is it's released by a neuron into a synaptic cleft. Okay, hormones could be the exact same chemical, actually. Right? We could have a hormone that's the same exact chemical as a neurotransmitter, but we would call it a hormone if it was released from a gland instead of a neuron, and it was released into the blood instead of a synaptic cleft. Okay, so if it's released from a gland in the blood, it's a hormone. Okay, so yeah, we have some examples of that where the same chemical could be placed in either of these categories, but they're just made in a couple different places and released in different ways. Okay, and then we have this one that's kind of a blend of the two, which is neurohormones. And neurohormones are released from neurons into the blood. Okay, so one example of that, of a neurohormone would be epinephrine. Epinephrine is what we call adrenaline, right? Our sympathetic nervous system causes adrenaline release, which is epinephrine. Epinephrine is released by our adrenal medulla, which is these little glands on top of our kidneys, which is made up of neurons, but they don't release into a synaptic cleft. They release it into the blood, and then it travels around our bloodstream and activates us, right? Epinephrine can also be released from a neuron into a synaptic cleft. In that case, we would call it a neurotransmitter. So it just depends on what releases it and what it releases into. Yeah, Jesse? Yeah, yeah, for sure. Yep, exactly. I mean, it could be either, really, right? Because epinephrine can be a neurotransmitter or it can be a neurohormone. In that case, it wouldn't fit nicely into one of these two categories. It's still the same chemical, right? So it's still going to have the effects that it would, but it goes into the blood, so it's probably closer to the neurohormone in that case. Yeah. Good question. Yeah, so that's common if somebody has like a peanut allergy or a bee sting allergy to have an EpiPen in their bag. Does anybody have one of those in their bag right now? Yeah, I got a couple. Yeah, yeah. What are you allergic to? Nuts. Yeah. How about you, just yeah. Anything? Yeah, we've talked about that before. Yeah. Okay. Yeah. So yeah, we'll get into that next semester in the immune system, but pretty much anaphylactic shock is what happens with that, and that's caused by all of your mast cells. All these cells of your immune system get activated all at the same time, and they release histamine. And histamine is a strong vasodilator. And so the way to counteract that is put a very strong vasoconstrictor into your body, which is epinephrine, right? And epinephrine does that exact opposite thing. It constricts all your blood vessels, so it's just trying to, and it's all about blood pressure at that point, right? Because if all your blood vessels vasodilate, your blood pressure plummets, and you can't sustain that for very long. So you have to do the opposite, shrink down all those pipes instead, and that's what epinephrine does. So yeah, have either of you guys had to use? Yeah, I remember you just recently we talked about it again. Oh my gosh, oh my gosh, yeah, yeah. Wow. How about you, Ryan? One time you said? Four times. Wow. Okay. Yeah, that's a lot. That's a lot. It's stress. It's a lot on your body when you have to do that too. It's not like, does it usually take you a while to recover from that? They give you some cortisol after. Yeah. Okay. Yeah. Wow. Wow. Yeah, so huge recovery process. Yeah, that's a big shock on the body. So yeah, all right. Thanks for sharing, guys. Yeah, so these are our definitions here of the types of ligands. Okay. When it comes to these receptors and channels, another thing to think about is what type of receptor is it? Okay. So we've been talking about ligand-gated ion channels. Okay. In this case, the receptor is also the channel, right? And so the receptor actually, when it gets activated by the ligand binding to it, it opens up, right? So it's not like a separate thing. It's like the thing that receives the ligand is also the channel, right? And so ligand binds directly to the ion channel and opens it, okay? These are very fast acting. They have their effect right away, but they're also very specific effect, okay? We can't have broad effects with these. This is what we saw like with the acetylcholine receptor on skeletal muscle that let sodium rush into the cell and cause that graded potential there. Okay, so that's one type. Okay, we can also have another type that uses has more of indirect effects and uses something called the second messenger system instead. Okay, so this is very common in the body. And so the ligand binds to a receptor. The receptor is not the ion channel, but the receptor still changes shape. But instead of it opening up to allow something in, it usually activates a little protein that's attached to it, right? So here's our receptor. Here's the ligand binding to it. Okay, activates this what's called a G protein. That G protein then goes over and activates another type of protein here, which then activates another one here, right? Which then does all kinds of stuff. It usually activates some enzymes that kicks off protein synthesis. Also will open up some ion channels as well, right? But we can have lots of different effects from this. This is all called this is what's called the second messenger system. Okay. So instead of the receptor opening itself, it activates a bunch of different events within the cell. You don't need to memorize these for this class, but what you do need to know is that this second messenger system leads to more broad effects, and we can have more effects in the cell instead of just that one thing, right? One of those effects can still be that an ion channel opens, so we can still get some depolarization or some hyperpolarization, right? But we can also activate transcription of some more proteins. We can affect some other things inside the cell. And so many channel receptors are this way. And it's probably the most common target for all the pharmaceutical industry are receptors that are connected to G protein coupled. They're called G protein coupled receptors. Yeah. Okay. Questions about that at all? Okay. Well, let's talk about some specific neurotransmitters then. Okay. So acetylcholine. We've talked about acetylcholine already. We've talked about it in motor neurons. Right? Talked about it in the somatic nervous system being excitatory for muscle cells, but it's also in the central nervous system. It's excitatory there as well. And then it's also in the autonomic nervous system where it can be excitatory or inhibitory. Okay. We find it in being released from the vagus nerve, which is the one that goes to our heart to slow down our heart to decrease heart rate. Right. So acetylcholine is the one that's going to do that. So that's just an example. Okay, we already talked about how it's inactivated, right, by acetylcholine esterase. It's broken down. Okay, and then there's all kinds of pathologies with it. We've talked about most of these. Talked about tetanus, talked about myasthenia gravis, talked about cure. There was a horrible chemical created and it was used in the Vietnam War, right? Called Agent Orange. You guys have heard of that? And that pretty much got in and destroyed acetylcholine esterase and also destroyed acetylcholine receptors as well. And it just led to permanent damage in people that inhaled it and that got into their bloodstream. And those were all related to acetylcholine esterase inhibition that caused. So that just led to way too much acetylcholine being out there all the time, right? So leading to too much muscle contraction, but then permanent damage in neuromuscular junctions as well. And then it's real common for insecticides like Roundup and things like that, other like weed killers and insecticides to use this different drugs that mess with acetylcholine pathways as well. So, Okay, dopamine is another one. Okay, dopamine is in one part of the brain that's really important for Parkinson's disease called the substantia nigra. Substantia nigra is a region of the brain that helps us kind of smooth out and regulate movement, and it has it plays an inhibitory role here. And so really it's kind of helping to smooth signals sent down our motor neurons. Okay. And so in Parkinson's, that's one of the main things that happens is these dopaminergic neurons, the ones that are in this part of the brain that release dopamine, start dying off. And so there's destruction of that part of the brain. And this leads to really jerky muscle movements, tremors, shaking, things like that you'd see and muscle rigidity, things you'd see in Parkinson's disease. So it's directly connected to the substantia nigra there. Dopamine is also in the frontal lobe, very important for mood, motivation. It's typically always excitatory in that case. Okay, this is all made from one of our amino acids, tyrosine, just a modified version of that. Okay, it's also known as a catecholamine. There's two other catecholamines in the body, which are epinephrine and norepinephrine. So dopamine is just the third one of these types. And yeah, it can be either excitatory or inhibitory. The other two applications here are schizophrenia. There's lots of kind of complex pathology to schizophrenia, but one of the things that seems to be known pretty clearly is that there tends to be too much dopamine release in the frontal lobe of the brain in schizophrenia patients, and that can lead to the mood disturbances as well as hallucinations, and kind of break from reality that is seen in schizophrenia. And then cocaine acts on a dopamine pathway as well. It blocks the reuptake of dopamine in the frontal lobe, and that's what can lead to hallucinations and that feel good kind of feeling with cocaine as well. So both those related to dopamine. Questions on those at all? Okay. All right. Go ahead and work through this one with the people around you here. You can put in more than one answer. Go ahead. Walk through that. Decide what you think here. Dopamine and Parkinson's. Check in with your teammates about this. Make sure you're on the same page.
That's just my guess.
All right, put in all the answers you think are right. Go ahead and hit the send button with this one.
I like this. I was really worried. So it'll give it a.
So nice, clear distribution. You guys just think it's all right. It's all got to be right. Okay, let's talk about that. So, first of all, let's go back here. So substantia nigra, dopamine is inhibitory there. Okay, substantia nigra is implicated in Parkinson's. Okay, we lose these dopamine receptors and dopamine itself. Okay, and so patients don't have the inhibitory input of dopamine. Okay, because it's inhibitory. And which of the two IPSPs or EPSPs would we not have enough of in this case? I's for inhibitory, IPSPs. Yeah, so because dopamine levels are decreased, that would not allow for IPSPs. So B and C here, right? So we'd be having less of this happening, right? That makes sense? Questions on that? Yeah, so because dopamine is inhibitory, that means that when dopamine binds to another neuron, it causes one of these. One of these IPSPs, this inhibitory postsynaptic potential. So without dopamine, we don't have enough of these IPSPs. We don't inhibit those signals enough. So it makes for those really jerky movements with Parkinson's or stiff, rigid muscles because there's just too much muscle contraction happening. So, other questions on that? Yeah, Sid? Yeah, good question. And so it all depends on the region of the brain that we're talking about, right? So it can, right? It can be excitatory or inhibitory, okay? But in this part specifically, it's inhibitory. Yeah. Great question. Okay. Other questions? Okay, so a couple more examples of neurotransmitters here. So epinephrine and norepinephrine. These were the two other catecholamines, okay? They're made from a modified version of that amino acid, tyrosine as well, okay? We have these all over. We have them in the central nervous system, very important in the brain stem, up in our prefrontal cortex is kind of like our most forward part of the brain, has to do with personality and important decision-making processes and things like that. But also in the peripheral nervous system, specifically our autonomic nervous system. We were just talking about the sympathetic nervous system and our fight or flight response. That's driven by the release of epinephrine and norepinephrine. Okay, so up in the brain, these could be excitatory or inhibitory, and kind of again feel good neurotransmitters. One of a very common antidepressants called tricyclic antidepressants. The reuptake of these is blocked by those drugs, okay? And amphetamines are another drug that blocks the reuptake of these neurotransmitters as well. So it just makes them act longer, right? If they don't get taken back up, then they stay out there and you have their effects longer. Yeah, those are kind of the basics on that one. Yeah, go ahead. Exactly. Yep. Math. Yep. Mhm. Yeah. That's it. Okay. All right. Another one, serotonin. So serotonin is a modified version of a different amino acid called tryptophan. And where do we get a lot of tryptophan? You guys know what food? Turkey. There it is. Yeah. So when you eat your big turkey dinner in a couple days, and you're feeling super tired after, they say that maybe it's connected to putting in a bunch of tryptophan that causes us to make more serotonin. It's not really proven though. It's probably just because you ate too much food and you're just tired. But yeah, what are you gonna say, Josh?
The serotonin turn into like melatonin.
No, they sound the same, but different. Yeah, melatonin is released by another little gland up in your brain called your pineal gland, which is released based on kind of how much light your eyes have seen throughout the day and other things, and that helps set your circadian rhythm for sleep and things like that. Yeah, so connected, but connected in just the way that they're spelt, but not the same. Yeah, yeah. So serotonin, yeah, in the central nervous system, important for sleep, appetite, nausea, mood, all these things. Definitely a feel-good neurotransmitter as well. And so this is the most common antidepressant drug that's ever been released, which is Prozac. Prozac blocks the reuptake of serotonin, right? And so then, yeah, depression may be due to low levels of these. This pathway is also messed with by LSD and ecstasy as well. And so, yeah, all different kinds of applications for serotonin. We also see lots and lots of serotonin in our GI tract. It's the primary neurotransmitter that leads to contraction of our muscles in our GI tract. So there's actually way more serotonin receptors and serotonin in our gut than there is in our brain. So interesting kind of connections at times between the gut and the brain because of that. So yeah, other questions on that? Yeah, Ethan? Mm. With the GI stuff? Not so much. Yeah. Yeah. Kind of a separate thing. Yeah. Because then with parasympathetic, most of those neurons are releasing acetylcholine. And then, so that is related to that. But then, sympathetic is all typically norepinephrine and epinephrine release. But yeah, some similar effects with sleep and things like that, but just different spots. But there is overlap here between why drugs like Prozac lead to nausea, is because it affects your GI tract as well. So, yeah. Okay. Other questions on that? Okay, so here's an interesting question to kind of work through. So ADHD involves problems with neurotransmitters in the brain, and it's treated with common drugs called stimulants, like Adderall, right? Adderall is an amphetamine, okay? And so why in the world would we give stimulants to people who are already hyperactive, okay? Talk about it for a second. See if you can have any ideas on that. Go back to maybe some of the neurotransmitters we talked about. And yeah, chat about it for a sec. And here, I'll give you a little hint on it. It blocks two things. So it blocks the reuptake of dopamine and the reuptake of norepinephrine. So then you can go back and kind of look at what those two neurotransmitters do in the notes and how would this help focus people that have a hard time focusing. All right, so let's chat about this for a sec. So this drug, Adderall, acts on two different pathways, right? So it blocks the reuptake of dopamine and norepinephrine, okay? Um, it's called a stimulant because of one of those two pathways, not because of the other one, okay? So the first thing it does is norepinephrine in the prefrontal cortex is inhibitory, actually, okay? And so that's not why it's called a stimulant, okay? That's the opposite of a stimulant. That's in inhibitory there, okay? So that's going to help with all this sensory information is coming into our brains all the time, right? And then we have to use the higher like executive function parts of our brain, like our prefrontal cortex, to kind of filter out and decide which stimuli are good ones to chase down and good ones to not chase down, right? And that's something that people with ADHD have a harder time doing, right? And they end up kind of focusing on all things at the same time, right? And so by leaving norepinephrine in the prefrontal cortex for longer, this helps us to inhibit some of those stimuli that we don't want to be spending our time on. Okay, so that's the inhibitory side. And then the reason it's called a stimulant is because the effects on the dopamine pathway, um, dopamine's excitatory in our frontal lobe and the motivation centers of our frontal lobe. So not only do we get help in filtering out stimuli that we don't want, that's from the norepinephrine side, but then we also get help in getting motivated to focus in on that one stimuli that we're trying to focus in on, right? And that's the excitatory part, the stimulatory part of it, right? So it's not necessarily makes people more hyper, but it gives them kind of hyper focus on one thing so they can really focus in on that and filter out the other stimuli that they don't want to be focusing on. So questions on that? Okay. My wife helped me with that one because she's a psych NP. So, all right. And then lastly, these two classes of neurotransmitters, endorphins and enkephalins. These are found in the central nervous system. These are pain relieving neurotransmitters. Their effects are mimicked by opioid drugs, so morphine, Vicodin, Percocet, heroin, methadone, all of these things, they block pain signals getting through our spinal cord and up to our brain. Okay, so these act more in the spinal cord typically, and those pain signals that are coming in from our peripheral sensory neurons, they actually never make it to our brain because these are the natural ones, these endorphins and enkephalins, but the synthetic ones like opioid drugs have the same effect and they just block that signal from getting up to our brain, so we just don't feel it. Yeah, Sydney, where you? Ibuprofen has a different way of doing that more by changing the inflammatory process, but yeah. Yeah. All right, guys, have a wonderful Thanksgiving break. Enjoy some rest, get some sleep, get some good food, and we'll see you guys next week.